Hello, can you hear me? Hello? Yeah. Yes. Okay, thank you. Welcome back. So today's session is data extraction and quality management. So hope you understand how to do the screening. Some of you have done, I am hoping so. So the next stage is after your final screening, how you want to do the data extraction. The data extraction is based on your objective. Like, for example, my objective is to understand the barriers. Then there are different types of barriers. For example, now we are doing PhD. So, publication is one barrier. Maybe fees is one barrier, or incentive is one barrier. Time is one barrier. Maybe sample is one barrier, reagent is one barrier. Like that, so many barriers will be there. So first we have to identify what are the barriers, and then we have to make it into an Excel sheet. So before starting your final screening, well, from the review like screening, like title and abstract screening, you will come to know the challenges or what are the barriers. So based on that, you have to do the Excel sheet. So this is same in case of systematic and scoping review. Based on your objective, you can frame. So one I will show you. This was a this was we are looking what are the challenges when we are developing a product in an industry from the APC till it reaches the market. That is my objective in this current study. So from this study, we have find out some challenges like formulation parameter like formulation stage, what are the challenges, analytical stage, what are the challenges, clinical stage, what are the challenges, then CPP related challenges, CQA related challenges, regulatory challenges, miscellaneous challenges. These are the challenges what I understand from the paper. So I make it into a headline and we just copy pasted whatever sentence in the final test, we copy pasted here. Later, when you're doing the analysis, this analysis is called thematic analysis. Thematic analysis, what we do is we will club all these two or three themes. For example, formulation challenges, CPP means critical process parameter. Process will come in formulation. Critical quality parameter, CQA means. This CQA is also formulation related. So what I will do is this formulation, this CPP and this CQA, I will club into one category. This formulation, CQA and CPP, I will club into one category, that is pharmaceutical challenges or for formulation pharmaceutical challenges. Now it is coming next is and analytical and clinical. We are putting into second set of challenges, that is bioanalytical. Only these two we are putting into second category, bioanalytical. Third one, regulatory and miscellaneous. Here miscellaneous mainly they are talking about the cost, how economic benefit and all. So here we are putting another category. It is called, for example, socio-economic and socio-economic challenges. So now from the data extraction sheet, I have nearly 7 type of challenges. Now I make it into themes. Themes is nothing but a common group. So one is pharmaceutical challenges. Under that, 3 type of challenges are coming. Next is analytical biopharmaceutical. In that, 2 type are coming. Then miscellaneous, like what you will tell, social demographic, it is coming too. Like this, you have to group this analysis. This method is called thematic analysis. So first you have to identify what are the challenges. Next, another one example I will show you. This one based on a guideline comparison we were doing. So here the main thing is I want to know, this is the first data extraction sheet. So here I want to know what are the different test. they are asking for particular drug. So these all are characteristic table. This characteristic table based on your study, it will change. If you are seeing any systematic or scoping review, you can see characteristic table. Like this is one article. You can see the characteristic table. In characteristics table, they will write the first order, country, aim, type of uh, product, subclass, what type of barriers you identify. This is a mini form of our entire data sheet. So like that, this is my first data sheet. In this selected columns are the characteristic column. You can choose country also. Then you can choose year of publication. You can choose first author name. Then type of article, like maybe some people use only research article, then not needed. Some people will use all research review, then you can choose that also. Everything you can choose. So here this was a guideline systematic review. So I choose drug name, their dosage form, then what is the route of administration, then which year the guideline was first published, then later which year it was. So this is nothing related to your topic. But here from F column, you can see this is what we have extracted from the guidelines. Nearly some 23, 24, 36 columns are there where we extracted the contents. So here, what are the studies required for the particular class of drug? That is our objective. So based on the objective, the type of classes, like type of test where in vitro was there, in vitro testing was there, then in vivo, some products have shown in vivo testing, then weight of evidence testing, qualitative sameness, quantitative sameness, physiochemical, particle size, rheology, zeta potential, like some, like you can see, I have mentioned, not mentioned, because if for this particular drug, it is not mentioned. Maybe so some other drug, it may be mentioned. So whatever is not mentioned, you can put it as non-mentioned. Then zeta potential, like this, so many. IVRT testing, IVPT testing, microbial testing, clinical study, pharmacodynamic, like this, so many testing are there in that final sheet, final extraction PDF. These are the tests. Some paper have that, some paper don't have. So now I have to do the thematic. I cannot represent 36 result in a single file or in a single paper. When you are doing the paper or when you are doing the data analysis, you will get n number of data. You will get some 72 hours graph. But what you will do, you will make it into a graph in such a way it will fit into a single paper. Like based on maybe time, we will not do 2 minute, 2 minute. If it is 72 hour, we will use some 10 hour, like that. Like that, this entire Excel sheet, we have to make it into a single paper report. So that analysis is called thematic analysis. So here also same like what I showed before, we have to do thematic analysis. We have to club some class of product like in vitro, in vivo, combined weight of evidence. All this will come under bioequivalent study. So here I have did that. Here I have, this is the second, this is my thematic analysis PDF, sorry, Excel sheet. The first characterization will be same. Drug name, drug dosage, route of administration, year of publishing, revised year, everything will be same. But from F column, my thematic analysis have started. In the first Excel sheet, you can see in vitro as a separate, in vitro as a separate column, in vivo as a separate column, and weight of evidence as a separate column. All these three tests will come under the class bioequivalence pathway. So I club it together and I make it into a single class. Second one, you have seen qualitative testing, quantitative testing, Q3 analysis. All three will come under the sameness criteria as per regulatory. Then next one, we already see particle size distribution, rheology testing, zeta potential. All these are individual column in my main data sheet. In my main data sheet, this particle size, rheology, zeta potential, osmolarity, pH, microstructure, all these will come in different different columns. Now I combine all these into one single criteria, that is physiochemical characteristics. So physiochemical, all these are the physiochemical characteristics. Those who belongs to Sutics, they will understand better. Now coming to pharmacology, if you are looking IVRT, analysis part, IVRT is a single column in my data extraction sheet. IVPT is single column. Microbial killing rate is single column. Dissolution is single column. All this will give you in vitro study report. So in vitro study report is the thematic analysis for this. Then in vivo, when you are looking, pharmacokinetic study is one, clinical endpoint, pharmacodynamic, then aqueous humor, like maybe ocular drugs are there, specialized studies are there. All these will come under in vivo study. So this analysis is going on, I don't complete. So this much from this, I hope you all understand how to do data extraction and how to do thematic analysis. This is the main thing what you have to write in your papers, and this is the way you have to do the extraction. Extraction, no one can help you until your colleagues who are working as a reviewer for you, because your criteria, your objective will be different. Maybe I am reviewing a paper. Same this paper, if you people review, you will look into different aspect. Now I am looking into regulatory aspect. If one person belongs to Sutics, they will only choose particle size. In particle size, they will look which which is the range, maybe which method they have used, or what is the acceptance rate, how much particle is counted. All these will become individual columns in your data extraction sheet. Maybe those who are working on IVRT method, which equipment they have used, which cell they have used, all these will come. So based on the person to person, if you are reviewing the same paper, the data will be different. So you should be very well known about the protocol first you have prepared. Secondly, your main objective, what you are planning to extract. So this data extraction can be done by a single person and can be rechecked by a secondary person, or you can share and do. The reviewers who did the review can share and do so that the bias can be avoided. This much only data extraction for systematic and scoping review. This is for systematic review what I have shown you. In case of I have shown you for scoping review. In case of systematic review, it is same, like what are the study, how much is the data, what is the percentage they have acquired, all this will come. How much is the particle size, that number, this is one systematic review thing. Here you see which apparatus was used, how much, all this like number things will come. If it is a research article, you will get all like 95% particle size. Another paper is telling 85%. That will come in these columns. If it is truck release, then you can say 85% truck release. Like that, the column will be. I hope you all understand how to do data extraction. If you have any doubt, you can ask this stage. Then we will move to data management, risk management. We will go. Anyone, any doubt in data extraction? Nothing. You have to read the paper. Before start reading the paper, based on your experience in the first and second screening, you can make an Excel sheet model. Then you can insert whatever it is there. If you are getting extra points, you can add extra columns and make your Excel sheet as wide as possible. After, once you complete your data extraction, based on the relevant, like first journal, like for example, now, if you are looking into this paper, most of the columns are not mentioned, not mentioned, not mentioned, not mentioned, not mentioned. So I can reject this paper for analysis if it is not giving relevant. But here I have accepted this because this point is more important when comparing to all other points. So that's why I have included. If you are feeling this paper is not that relevant, then you can reject. But if most of the columns are filled and you are feeling still that paper is not relevant, that means some kind of um, misunderstanding was there. So you have to recheck again. Now let me know any doubt in this stage. Hello. Hello. Should I explain once again? Please let me know. Okay. So now you have got final extraction. Some, for example, you finish your data extraction, title and abstract, you will get final some 32 papers. Out of that 32 paper, now you have to do the data extraction. So what you have to do is like this one Excel sheet you have to make. This is your data extraction. If you do it in Ryan also, if you do it in Excel also, it is same. But the main problem related to Ryan is once you do your data extraction in Ryan means you cannot copy paste into an Excel. Again, you have to sit and type in the Excel. So what we usually do is we will do it in the Excel sheet. So once you get these things, then first you have to make a 5 to 6 characteristic column. Characteristic column means you have to look into the type of study. How we will usually write in paper is when it comes to the result portion. Okay. When it comes to the result portion, we will write like which day we have did the search and what is the search approach we have used. That is your Prisma guideline. Sorry, Prisma, not Prisma, your search stream. That search stream number of article you will be noting here. And then you will write about the Prisma diagram. This many articles was found. This many duplicates was there. Then title and abstract screening was done. From that, this many article we got. Again, we will do full test screening. This much is your primary data. After that, when you write the result, you have to write the characterization of the included study. Characterization of the included study is nothing but now after final screening, if you get 30 paper, you have to write a characteristic of this table. So here, now the characteristic what we have written is from here it was starting. So most of the studies were conducted in Europe, US and India, China, Russia, like that. How many paper we have noted in blue color? That is one characteristics. Second characteristics is our study design. So we have chosen review articles followed by research articles. Some people only choose research article. So you have to write the characterization study design like cohort study, case control study, then observational study like that you can write here. So since I have taken review and research, we have noted like this. If you are taking review, then you can write like systematic review, how many articles, scoping review, umbrella review, how many you can write. So study design as a characteristics. Third one we have noted what are the formulation type. Since it is one Sutix base, we have mentioned the formulation type. So complex APIs are there, drug device combination is there, like that. So this is your characterization. Till here it is characterization. Then what are the barriers identified? This is the first Excel sheet what I have shown. So formulation is one barrier, analytical is one barrier, clinical is one barrier, critical process is one barrier. So this is the main characteristics of the study. So when you are making your Excel sheet, the first 3-4 columns should be the characteristic column. Sorry. The first 3-4 will be the characterization column. Here I added the author name, country name, anything you can do, but you have to explain in the result portion. Next, from column D, I have column F, I have made the objective. My objective is to understand the barriers. So based on my observation, I find out these are the barriers. So each barrier I have pasted here, but this big Excel sheet, we cannot put it in the main study or as a result. Now, why you are doing systematic and scoping review? You want to know which is like scoping review means you have to know which is the scope. Now you are seeing formulation having more challenges means you will work on formulation in order to overcome the barriers. Then you will get more chances there. So you are looking the scope. So to understand the scope, you have made the broad one. So now you have to find out a theme, like formulation is also higher, CPP also higher, CQA also higher. All these will come under one category, pharmaceutics. So that is categorizing the things. In seventh, eighth standard, we have studied no, uh, like kingdom animalia, kingdom plantae. Under that, so many classes like that. Here also we are making a categories into a single theme. That analysis is called thematic analysis. Usually for systematic and scoping review, we will do thematic analysis. So once the thematic analysis is coming, like for systematic, it will come numbers. So when it is looking some differences, like we can compare something, then we will go for meta-analysis. This much only, the main thing. Here, the second one, what I have shown is this is my data extraction sheet. It was a very vast. You can see how vast it is. So this data extraction sheet, we have minimized into some, sorry, this much till here. That means column M. Actual data extraction is column AP. So that column I have made into column M. Now the result will be this much only. This I can represent in a single paper. I hope you understand. And we cannot tell what criteria we have to put it in the Excel because it is depends on what objective you are working on. Okay. So next we are moving to risk of bias assessment. Risk of bias will be there always. So what is risk of bias means? Already when we do screening, I told you there should be 2 reviewers. Even though there is 2 reviewers, 2 reviewer opinion we cannot consider as a generalizability will not be there. If you want to generalize, like for example, if you want to generalize, okay, PhD is easy, then in India, you have to do some 100, 200 sample size. You have to get their opinion. In the same way, you are doing reviewing, 2 of the reviewers are doing, and then they are telling, okay, these are the main barriers. No one will accept you. With this data, you cannot do a systematic and you cannot do a meta-analysis. Mainly, this is not applicable for scoping review. Risk of bias for scoping review, if you are looking, yeah, if you are looking into like systematic and scoping review, here Prisma SSCR checklist is there. Prisma SSCR checklist. You can search in Google, you will get the empty checklist. Then you have to just report like where you have placed the title, whether structured abstract is there, then whether you have mentioned rationale of the work, whether you have a place objective, then protocol registered. It is not necessary that you have to register the protocol for a scoping review. For systematic, it is mandatory. Then eligibility criteria, information. If you have did all that, if your column is full, not or almost full, then the checklist is fulfilled. Now, if you are looking critical appraisal within the source of evidence, I have not done. So I have put not applicable. So this checklist along with your paper you have to submit. If you don't submit, no one will consider your paper. So this format you will get from net. If you want, we can mail you also. This is for scoping review. For systematic review, you have to do the risk of bias. This critical appraisal is of evidence is risk of bias, but you can do for scoping also, but it is not mandatory that you have to do. Without this one, you can do. So coming back to risk of bias assessment in systematic reviews, that's why I wrote here systematic reviews. There is no scoping review, risk of bias. So why we are doing risk of bias? Because to validate the general ability. If you are telling one thing, it should be generalized means some people have to agree. So to general ability and to pull out the estimates, like some people, if you are chosen 30 study, in out of the 30 study, with 2 or 3 best study, you can prove that my hypothesis is correct. So to pull out such studies and do the meta-analysis, we are doing risk of bias. So here, this risk of bias versus quality assessment. So what is the difference between risk of bias and quality means? Once, like you are checking the risk. If I am doing so, any risk is there. Okay, if risk is there means it should be high quality. Now we are giving medicines to America. Then medicine quality and what we are getting in India is different. Why? Because if it goes out, if it retract from that country, then it will be a risk for us. So risk is always related to quality. So risk of bias is like if you get a better risk of bias format, like your risk of bias is null, that means your quality of screening was good. If the risk of bias is not that good, the value is not that good, means your screening is not that well. So it is like a validatory method to look whether your analysis is good or not, whether your analysis is bringing some impact to the society or not, or whether you have correctly reported the data or not. So risk of bias, mostly this Prisma, Cochrane and all, they are mainly focusing on ROB. This software is a very vast software, how we are doing this coursework. If we want to do this risk of bias, you have to do another one big coursework. It will be some 7-8 sections it will be needed for proving this risk of bias assessment. Since we are not dealing with meta-analysis in this coursework, we are not going in depth. For a systematic review, how you can easily publish with risk of bias, we will teach you. So risk of bias tools, as I told, there is many tools, that is ROB2, ROBINS, Newcastle, Quadas, then SBI critical appraisal tool. All these are tools which are used for risk of bias assessment. This ROB is the main software. Under this, these many sub tools are there, which is used for the analysis based on that study. If your study is a randomized control trial, you have to go for ROB2. Then Robbins if it is non-randomized, Ottawa if it is case control coherent. So based on these studies, you have to select the common tool. So here we will give you an outline of how to choose this softwares for your meta-analysis. So first one ROB where you have to do is randomized trials. So in randomized trial, when you are making an Excel sheet, there should be mainly 5 domains. What is the process of this is what you have to include in your Excel sheet, because with you can add n number of columns, but this 5 columns should be there in your Excel sheet when you are doing a randomized trial procedure, RCTs. So in that Excel sheet, one column is what is the process of randomization. Then second one, any deviation from the initial intervention. Then any missing outcomes are there. Then how they did the outcome measurement and selection of reported. I will share you this slide. This thing should be in your Excel sheet plus extra whatever is needed in your study. So next is Robbins method. It is for co-founding study. Co-founding study means, for example, I am telling those who doing exercise, they are having good health. So your main subject is exercise. Having good health is another one criteria. But the one another person is telling, okay, you are doing this exercise and getting good health means people who have money, they are going gym and doing the regular exercise. So money is a factor, or their socio-economic background is a factor. Correct? No. Otherwise, you can tell. Uh, okay, there is some problem in the house because her father is staying here and mother is staying some other place. That means father and mother staying is different place is one criteria. Problem is one criteria. The third thing may be the father is a drunken person. So that is the third party, what it is playing in between these two. So there is two things, but a third party having an influence means that is called co-founding study. So such studies we are using this one. So in that case, you have to have what is a participant selection, whether it is a randomized one or control one, or you are choosing randomly or not. All this will come. Then intervention classification, then missing data outcome. Next one, Ottawa study. Here it is like a, you have seen in some systematic review, you can see some star mark like our stars in the sky. You can see some diagram with star mark. Usually it will represent cohort studies. So here, like for example, when you are comparing smokers with non-smokers, then you have to use all these column. For example, selection of first participant. What is the selection criteria? What is selection of second participant? What is their exposure rate? What is the outcome? All this should be there. And for these things, we are giving star marks. We are not assigning any points. And last, we will count the star. And if it is above 7, it will be a good one. Good one means good quality one. Good quality means indirectly proportional. That means low risk of bias is low. Risk is low means it will be a good work. So like that it will be. So this is mainly for cohort and case control. Usually pharmacy practice people will use this. They will also use mostly ROB2 domains also. Next one is PRODAS. Here this is like what you will tell is it is a bias. You have some kind of like one person having brain tumor. They are taking MRI. First we will take MRI. Then they are going for biopsy. No one will directly go and do biopsy because we have to first confirm. Then we will validate. So there will be some bias. Which one to do? Why I should waste money for MRI? I can go directly to biopsy. So there is some kind of bias between the things. Then we can use this kind of risk of bias assessment. So these are the parameters there you have to index text means like a primary test what we have to do. Like if you are comparing biopsy with MRI is the preliminary test what we will do. So that is index text. So this column should be there and your judgment here is not based on star mark. It is based on low, high, unclear. You can see some diagram. Example. You can see like this some diagram plus minus low medium here it is written no low high or you can use like this diagram. This kind of diagram are seen in systematic scoping review. This is based on that analysis. Last one, JBI critical appraisal tool. This is just similar what I show here. Like this is the JBI form. This is for systematic, sorry, scoping review. Same like this for systematic review. Another checklist is there. You can download the checklist and do if your study is a cross-sectional or other studies other than this RTCs and cohort case control. If other checklist studies or questionnaire based study, interview based studies, or some guideline comparison and all, if you are doing systematic review, you can use this tool. Then traffic light visualization. Traffic light visualization is nothing but what I shown now. These are the main tools actually. So risk of bias is nothing. We have to look the validity of the study reporting, how we have did the screening and all. We have to look whether it is correct or not. So we are doing the risk of bias. So risk of bias versus quality. If risk of bias is low means the quality of your work is high. Just remember risk of bias and quality is indirectly proportional. Then, which are the main tools? Based on your study, you have to choose the correct tool. So many people have mistakenly used JBI critical appraisal and they will send their RTCs and it will get rejected. Seeing the study only, people will understand. Within the first review cycle itself, people will get rejected because of the wrong selection of the tool. So be careful. This JBI tool can be used for cross-sectional survey or cross-sectional interview studies, case reports. Usually I am using JBI because I am doing cross-sectional studies. But these are the advanced one. And in our college, they are using this one, third one and first one. Other type of studies are not that much here. Mostly it will go in medical MBBS type. Then this ROB, what are the columns you have to include in your Excel sheet? These are that. I will share you this one. So next is representation. Now, any doubt in this? What are the tools? How to do, like not how to do, the basics of risk assessment. Because for our publication of simple systematic review, we don't need this risk assessment. For meta-analysis, you need this. So if anyone wants, you can just message us. We will try to get some experts in this field and we will do another one coursework on this topic. It was a vast topic. You can see so many YouTube videos, but nothing is that much detailed videos are there. No, nothing like that. Animal and preclinical study, no special tools. It will be depend on the papers how you are. Now you are taking a cross-sectional based animal study. Then it will go, of course, in a JBI tool. And if you are going for an randomized or any case control, like you have a test group and control group, you are doing testing in animal and then it will go in Ottawa type. Depends on your paper when how you are extracting. Only this many. These are the only classes of tools, nothing else. And these are the type of studies. No more tools. As of my knowledge, this much only. And recently, I don't know any other tool scheme. This, if you want, I can help you with ROB and JBI. Now we will go for representation. So this much only what is the tools. So next is traffic light visualization. What I have shown you like that only. This is only traffic light visualization. You want to create a traffic light visualization tool. You can see here, these are the traffic light faults. So when you are representing in your systematic review, you can mention that figure legend as traffic light plots. It can be any of this. Usually people will use this first one. So that is the way we are showing. This is just to summarize your result, whether your result is good or not. I show you now one big Excel sheet will be there. So again, this Excel sheet also, if you are going, this is one of our study, what we have developed this traffic light. Actually, this much like 25 columns are there. So again, 25 columns representation, your paper will also have 25 columns. Even you reduce the width of the column, this lengthwise it will be high. So what we will usually do is, we will just write the title like first author and year. Then we will like formulation challenges, less explained in the paper. So I wrote low. Analytical challenges in this paper, everything is telling that these all challenges are very low. So I put low. Then in this paper, it is unclear, not clearly mentioned, but all these are low. Maybe in this paper, everything is high. So we are only looking whether the challenges is more influencing our study or not. If it is high, then you can mention high. If it is low, you can mention low. So you have to create like this for the 25 study. This I did for a rough analysis for you. So we have to do like this, and we have to check whether our challenges is low or high, highly impacting our objective or highly impacting the society or less impacting. So you will get a Excel sheet like this. From this Excel sheet, you have to draw the diagram like this. So for drawing the diagram like this, how you have to do is, you have to go to Rob visualization tool. I think you can see my screen. Yeah. So this is the Rob visualization tool. You go to this site. There they have just explained how to draw and all. Here you go to Rob's tool. So you have to go to Rob's tool. Here how to do settings and all is there. Here so many data sets are there. You can download this data set. If it is your study is a Rob 2 type of study, you can download this data sheet and make your study in this format. In this format, D1, D2, D3, D5 overall, and study 1. Instead of study 1, you have to write the first author name and year. So I have did a general, like what you will tell, crossover study. So I just download general data sheet. The general data sheet will be like this. Some sequence, something they have written. So I changed this one into my criteria, formulation, analytical, and I am writing my first order names. And 2020, sorry, 2020. I have to rearrange. Maybe my first study have told very nicely about formulation. It is highly impacting the production. So I am feeling it is a high. So like that you have to type. Then you have to upload your data here. When you upload, you have to choose. If your study is Rob 2, you choose as per Rob 2. Since my study is generic, I choose generic browser. Then I have already did mine. So this is the Excel sheet I have prepared. So just upload it. So you can see the Excel here. Look, my first author, second author, third author, like that 25 authors. Then their challenges, how they have explained about the challenges. Overall, what is the score I got. Then put generate plot. Once you put generate plot, all these criteria will come. Same. I have pasted 10 first 10 people, so it comes 10. Then 12 text patters are there. Everything it will come. Here, if you want to change anything, now it is low. I want, instead of unclear, I want medium challenge. So I put medium, then high. So this diagram you will get, you can export here down. Sorry, here you can download the plot and you can use it for your publication purpose. This is what you are putting in your risk of bias analysis. If you are using ROB software and all, you can get this plus minus easily. Like easily in the sense it will take some time. You have to do again analysis there. Otherwise, one thing you can do, you can take JBI format. You can check whether all these criteria is mentioned in this particular. Usually for symbol review, we do like that. We will look whether this paper is explaining more about the study. Whether this paper is explaining more about the study, maybe objectives are similar, the participants are similar, or the result or our hypothesis and this is matching or not. All that parameter we will see as per JBI checklist and we will write high, low, unclear and all. And just we upload, I hope you understand. Just to go once more, I will tell, go to this ROB website. This is called ROVIS for visualization. Just to click there, go to ROB tool, then upload your file there, upload your data, then choose whatever it is the study type, choose it, upload your data sheet. Once you upload, you just make sure you have uploaded the correct one. It will be in the left hand side, then generate plot. It will show the traffic plot for you. So this much only the risk management and risk visualization. If you want to go in depth into risk management, then all the tools we have to show you how to use, but that is necessary for only for meta. If you are wishing to do a meta analysis, if your data are good enough to do a meta analysis, if your data are not good enough to do a meta analysis, you can stop your systematic review here. And it have value also, no issues for if you don't do meta-analysis also, it is will be acceptable. Okay, any doubt? This much only today's session. This is my last class. If you have any doubt related to this, you can ask me now, or we'll be meeting in the final discussion. And if you want to know more about this risk management, you can search in YouTube's and all. Otherwise, in the final discussion, you can ask Rashi sir also because he have used so many risk assessment techniques. Usually, pharmacy practice people have used these RCTs and all more. And if you wish this meta-analysis, you can contact us. We will take in depth about risk management. With this, you can publish a systematic review. Any doubts? Anything should I explain? Yeah, just please type even Lakshmi, Kirtana, Bhumika, Navas. Whether it was useful for you, anyone, anything, let us please know. Okay, then bye. See you all for the next class. Bye.